My name is Dr. Msomi. I'm a clinical virologist. I am based at the Albert Luthuli Virology Lab at the University of KwaZulu-Natal, also part of the NHLS, which is the National Health Laboratory Service, which offers a diagnostic service for all virological diseases to the public health sector. I am the head of department, and in my department we offer undergraduate and postgraduate teaching and training of people in the field of virology, specifically in the diagnostic virology field. At undergraduate level, we teach undergraduate medical students, biomedical science students, as well as any field that would use virology as part of their practice. And at postgraduate level, we've got different professional qualifications, from specialists who are medical specialists to medical scientists, as well as medical technologists who offer the holistic service in diagnostic virology. So the field of virology have different areas or functional areas. We diagnosed any viral diseases using different methodologies, serological methods of diagnosis, where we use patients' immune responses to viruses, and that is what we call viral serology. We also use molecular methods of diagnosis, and that has become the prime focus of our diagnostics in more recent years. Traditionally, virologists also do culture viruses, and I'm still old school, so as you can see, I still culture viruses, and we use microscopy to see the effect of viruses in cells and in the human body. In our viral serology lab, a whole array of viruses can be detected by using what we call immunoglobulins, IgMs, IgGs, to see if there's been exposure to viral infections. On the molecular side, though, because viruses by their nature are just nucleic acids, DNA and RNA, that makes it the prime target for diagnosis, and that is why that's become the cornerstone of viral diagnostics. And so most of our viral diagnostics center around what we call now nucleic acid detection methods, an example of which is the PCR method, and that is what we use to detect signature sequences that are unique to each virus to identify a virus, and that is how a diagnosis is made. We also use molecular methods not just to diagnose, but also to monitor patients who are on treatment. So patients who have been on treatment for a while and we want to check if there's been response to treatment, we would do what we call monitoring virology, and we'll do things like viral load to see if the virus is responding to the treatment by reducing or increasing, and that's how a doctor would act on how to manage a patient. On the research side, we also use molecular methods to sequence viruses, especially with new and emerging viruses. This has become a very crucial diagnostic method because in that you can make a unique sequence of each virus and identify if it is something that's already known or if it's something novel. An example is SARS-CoV-2, and our lab was quite instrumental during the COVID-19 epidemic and setting up a new diagnostic system for a novel pathogen for which we did not really have existing diagnostic methodologies. We use sequencing now also to monitor in response to treatment to see if there are any emergence of any variants or if there is any drug failure. We can use that molecular method of diagnosis. And also on the discovery side, we still go back to old school ways, which is growing viruses in culture and seeing how the viruses behave, and that is how we can see if viruses attack particular cell types, particular organs, and that helps with discovery because you can also see if a virus in culture, which is how it would behave in a human body, would respond to different modalities of treatment or vaccines and the likes. And that is an area of research that I'm particularly interested in, using cell culture-based method to find new ways for approaching chronic viral infections and viruses that tend to cause cancer. So my area of interest is in viral hepatitis. Hepatitis B specifically is one of the viruses that causes cancer of the liver, hepatoma, and my research is to try and eliminate the burden of hepatitis B as a public health threat by controlling response to treatment and by monitoring how it becomes resistant and things like that. There are many different areas of research. My other colleagues also do HIV research and many other viruses. I know we have the popular viruses like HIV and SARS-CoV-2, but viruses, there's quite a lot of them. And in the department, we do have an ability to make a diagnosis of any of the viral diseases, viruses that can cause viral diseases of public health significance.